Okay, now let's go into looking at some of the crystal structures to kind of show you how these things work. And beyond the sort of general idea of how these things work, I don't think there's anything specific. You're not going to have to like draw these structures out from memory. Yeah, that's a bit too much. So look at the molecule over here on the left. Can you recognize the parts? Don't even look at the part at the right that tells you what's what. What do you see here? So I see a beta 2M right over here. And I know beta 2 microglobulin is only found associated with MHC class 1. So this is going to be class 1. And look, there's my floor, and there's my alpha helices that are like the little binder clip. And so this yellow thing that's running through there, that must be the peptide. And look, the peptide is nice and short, which is also right for an MHC class 1. Now, what could be binding to an MHC class 1 with a peptide on it? And the answer is a T cell receptor. That's the thing that binds it. And so our T cell receptor up here is going to have these little hypervariable domains. That's the red, blue, green, yellow, and purple over here. And it looks like this hypervariable domain over here is not getting used in this particular one. And together they are interacting with every little bit of this peptide. So this is how a T cell actually recognizes a peptide. The peptide has to be stretched out and locked into place by MHC class 1, and then the T cell receptor can interact with it. Now, can you remember, if this is an alpha beta T cell and it's interacting with MHC class 1, is it going to be a CD8 T cell, killer T cell, or is it going to be a CD4 T cell, which is a helper T cell? Think about it a sec. Did you get the answer? It's going to be a CD8 killer T cell. They are the only ones that interact with MHC class 1. And they're going to do it with not just the T cell receptor, but a second molecule that's going to reach around. It's a big, long molecule, and it's going to reach around the side and actually hold on to both the T cell receptor and the MHC class 1 at the same time. And so that's going to make sure that we have good contact there. All right. This is an MHC class 2 binding. And yeah, same sort of thing. So look at this without looking at the legend over on the right side. And I think you can maybe actually start to recognize some parts of this. So up at the top, we got a lot of immunoglobulin-like folds. And so that could be a number of things. But at the bottom, we've got two immunoglobulin-like folds, and the yellow one is part of this half. Look, there's the floor, and here's our big yellow one side of the clip. Here's our other brown side of the clip. And so this is going to be MHC class 2. And you can see there's a peptide in there, but this side of the molecule is open, and the far side would also be open, although you'd have to rotate that around to be able to see that. And so what is it that interacts with MHC class 2 that's bearing a peptide? The answer is going to be, it's still the alpha beta T cell receptor that's going to interact, but we're going to need an additional molecule. Do you remember which one it is? It's going to be CD4, right? Because what we've got here is the interaction between a helper T cell and some other cell, something like a dendritic cell, would be presenting little peptides to a T cell in order to help educate it, help show it what to make antibodies against. Or what to help B cells make antibodies against, rather. All right. So CD4 and CD8 proteins are the cell surface proteins, and they're the ones that are going to reach around to the side, and they determine which MHC molecule you can bind. If you have CD8, you can bind MHC class 1. If you have CD4, you can bind MHC class 2. And that's it, and you have to have a T cell receptor plus CD4 or CD8 in order to have binding. So CD4 and CD8 are called co-receptors. They aren't the thing that's actually binding to the peptide, but they are necessary in addition to a T cell receptor to get binding. And so T cells are going to have either CD4 or CD8, and B cells are going to have a complex of 3 proteins, one of which is a complement receptor, but we'll get to B cells and their receptors shortly. Okay. So then there's going to be LCK, which is a kinase. And a lot of these kinases are going to end in a K. It's going to be 3 letters, and often you would see this as, if it is the gene, it would be LCK lowercase and in italics, and if it's the protein, it'll be LCK uppercase, either all uppercase or sentence case, the way you're seeing it here. You see it both ways. And yeah, this is one of the kinases that interacts with the tails of CD4 and CD8. And we'll get on to those diagrams, I think, pretty heavily in Chapter 5 as we sort of explain how this works. But the stuff that you're building right now is going to pay off. It's going to totally pay off in Chapter 5 and Chapter 6. Just watch. Okay. And so in addition to those normal alpha-beta T cells, which are the ones that have the CD4 and the CD8 and act like we would normally consider T cells, killer and helper T cells to act, you're going to have these mucosal-associated invariant T cells, which are going to be mostly gamma-delta T cells with, very limited diversity. And they are going to be, they're going to respond mostly to bacteria. And so we will get to those as well. MR1 is a non-classical version of an MHC class 1 molecule. And it's a version that pretty much just binds certain metabolites, certain things that a bacteria has changed folic acid into. So this is kind of like a bridge between innate immunity and adaptive immunity. These MAIT cells, which are mostly gamma-delta cells, are going to be found on mucosal membranes. So think like your intestines or your lungs, something like that. And they're going to have, instead of a normal MHC class 1 molecule they interact with, they're going to interact with this MR1. So there we go. And we'll get on to more of those later on in immunology. I think we mostly focus on alpha-beta T cells in this particular chapter. So your CD4 molecule looks like this. It's one long chain, and it has 4 of these domains, and I think they are all immunoglobulin-like domains. Oh, yeah, look. 1, 2, 3, 4 immunoglobulin-like domains with a couple of alpha helices, that's these little cylinders, in between the domains, but for the most part, we're just straight-up IG-like domains. CD8 is going to have 2 copies, an alpha and a beta chain, and each one of those is going to be an immunoglobulin-like domain, and they're going to have a little helix over here on the side of that domain, but it's the main domain that we use to classify these. It's like an immunoglobulin domain with a little extra on the side. Look at the way they interact with their respective molecules. So here's the CD4. It's going to be coming from the T cell, and it's going to interact with the MHC class 2. So something like a macrophage maybe could present with class 2, MHC class 2. It's the white blood cell only special type of MHC. Here is CD8 molecule. And so once again, this is coming from the T cell. And it's going to bind to the side of the MHC class 1 molecule. You can see the beta-2 microglobulin here in pink. Whereas the whole alpha chain is shown in pink over here, but that's equivalent to the equivalent to the beta-2 microglobulin and half of the antigen binding site at the top, the antigen binding groove. So what's missing from this picture is going to be the T cell receptor, which is going to bind right up here, sandwiching that little peptide and this little MHC class 1. Or over here, it'll also be the T cell receptor, so probably that alpha beta T cell receptor. And then this CD4 is going to be presented on a long chain, so we're missing the rest of the chain here that's going to reach down to the T cell. And we're missing the rest of the CD8 chain. That actually hasn't been solved yet. We don't know what the structure looks like. We just know about how long it is. It's got to be long enough to reach out to the side of this And yeah, maintain that interaction, which is what we need. So here is now the entire complex. So here's your alpha beta T cell receptor with the alpha chain in red and the beta chain in blue. Here is MHC, class 2 molecule. So here's our 2 alpha helices at the top, and they're going to be pinching a little peptide right in the middle, which is either this gray thing or is not shown. I'm not sure. And then over here, you've got your CD4 molecule, which is going to be reaching out. So the T cell is going to be up here at the top, and the cell that is presenting to the T cell will be down here. So something like a macrophage. Okay, CD8 is going to bind to MHC class 1. And yeah, it binds to a different part of the MHC class 1 receptor. It's going to bind to the part of that alpha chain here. I think it's the alpha 3 domain of that. So it's not going to bind to the beta 2 microglobulin. And so, yeah, that's fine. And again, your T cell receptor alpha and beta is going to be at the top. So the alpha is going to be this sort of, I don't know, lighter color, and the beta is going to be the darker, more purpley color. And there's CD8 reaching around. And you can see it's pretty much doing the same job as CD4. Here's our alpha beta, here's our MHC class 2, there's CD4 reaching around, alpha beta, class 1, CD8 reaching around. So they're analogous molecules, and they're doing similar jobs, but they allow different subsets of cells to recognize a peptide. And they can recognize different peptides between class 1 and class 2. All right, so who has class 1? Who has class 2? Let's see. So, T cells are actually going to have class 1 so that they can show other T cells what they're making. This is important because if you have a T cell that gets infected, like in HIV, for example, it needs to be able to present little peptides of that HIV proteins so that that T cell can be recognized as infected and dealt with. And you're going to get class 1 on, yeah, it ends up being on most cells, like kidney cells, brain cells, liver cells. Pretty much everybody's got a little bit of class 1 on them, except red blood cells. And red blood cells aren't even proper cells. They don't have a nucleus. They're just a fragment of a big cell. And red blood cells are kind of already on the way to dying. They just haven't quite died yet. That's what they are. That's how we use them. There are a few animals where they're actually living cells that still have a nucleus, but not in humans. Not in us. So yeah, MHC class 1, pretty much everywhere. MHC class 2, though, only found on these lymphoid tissues. So T cells will have a little bit, but B cells and macrophages and dendritic cells and epithelial cells in the thymus, so this is going to be a sort of a T and B cell education zone, can also express a bit of class 2. And so even the walls of the lymph node effectively are going to have a little bit of antigen presenting capability. So in humans, activated T cells are going to express class 2. That's what the little star is for. Whereas in mice, T cells are always class 2 negative. And so a lot of what we've learned about them is from mice. But yeah, the positive does refer to us. And so I think that's a fair thing if you're just memorizing some of this. All right, so those are our classical molecules, our classical MHC class 1 and class 2. And they're going to bind to the T cell receptor with the peptide in between, and they're going to have either CD4 or CD8 reaching around to the side of them. We also have non-classical MHC molecules, and they're going to be called class 1b molecules. These are very restricted, and like I said, they're going to recognize mostly different versions of folic acid that have been modified by bacteria. So it's just a different way to recognize certain bacteria. And yeah, transitional immunity is going to be recognizing non-peptide ligands, which are non-peptide antigens, if you like. And it's mostly going to be done by these MAITs or gamma delta T cells. So they're they're a not quite as smart, and you could even, it's the adaptive immune system, but it's the least adaptive wing of the adaptive immune system. So we think that these may represent a more primitive kind of transitional state. So as we were evolving toward what we have now, this adaptive immune system, we think we may have gone through a stage where gamma delta type T cells and MAI-Ts were the only kind of T cells that we had. Okay, and then T22 is going to be a mouse version of a class 1b non-classical MHC. So if you see that anywhere, that's what that is. And it's going to be recognized by a subset of gamma delta T cells. Okay, so what's it actually look like? Well, glad you asked. So here, since we have a T22, you can tell we're in a mouse, but this is going to be one of these non-classical presentations. So we're going to have a beta-2 microglobulin, IG-like domain, immunoglobulin-like domain, and the T22 molecule is going to take the role of the MHC class 1. So this is our class 1b, and pinched between the alpha helices up here, we're going to have a little molecule, and it's always going to be some version of the same molecule, that folic acid metabolite. And then the things that interact with this are going to be the T cell receptor gamma delta version, and that's going to pinch it and form basically the same sort of structure. But this is what you're going to find in those gamma delta T cell interactions. Okay, and that is the end. Yeah, those two sections where we went into the details. There's nothing in the particular details of those molecules that we need to memorize, because with different variants of MHC class 1 and 2, you will get different binding, or with different variants of the T cell receptor, you'll get slightly different binding patterns. Those are just good examples to give you an idea of what to expect. All right, and now you know about the structure and the way that antibodies and T cell receptors can bind to their antigens and present them or recognize them. And that's big. This is the nuts and bolts of how the adaptive immune system actually works. And we'll go into the genetics of how we actually get these immunoglobulins and T cell receptors to be assembled in the next chapter. But that's a very chapter 5 thing, and for chapter 4 we are done. So I'm hoping the second half of this was a little easier because it built on the concepts from the first half, but you tell me. Yeah, I'm interested to know how this worked for you.